Good morning, ladies and gentlemen. It is my pleasure to be with you one more Thursday. And today we are going to be hearing from Dr. Jason Mighty, who is related to Stephen Mighty, who we all know. And Dr. Mighty is of Jamaican origin, but he did his BSc, Master's, and PhD at colleges in New York. So he did his PhD at the City University of New York, the Graduate Center. That would have been in May of 2019, but he has had a lot of experience in other, prior to getting that degree. He has worked with the Deerfield Management in New York, Simon Kucher and Partners in New York as well. And he is currently with Regeneron Genetic Center in New York. And there he supports the scientific operations team in facilitating large-scale human genetics projects, identifying and validating potential drug targets. So you can see that this is very, very important and critical work. He has publications in the neuroscience and oncology fields. And he also, during his PhD, he was a program coordinator for the Louis Stokes Alliance for Minority Participation, that's LSAMP, at multiple CUNY campuses, where the focus really is to increase engagement of minority students in STEM research and pursuing graduate careers in the STEM field. So you can see that clearly he's a motivator and he has those skills and abilities. He has done a lot of traveling, and that includes being in Austria and the Netherlands. I trust that he will speak to us about some of those experiences that he has had at those particular locations, as well as maybe other places that he would have been able to visit. So the title of his talk this morning is A Mighty Journey Through the STEM Field, and I am going to just go right ahead and hand over to Dr. Mighty, who is going to be sharing with us this morning. Thank you for joining us, all persons, and thank you for joining us, Dr. Mighty.
Oh, thank you so much. Appreciate it. That was a hell of an intro. I feel so special now. Awesome. Good morning, everybody. Thanks for having me. Appreciate the time. So I just want to talk to everyone today about, a lot of things I've gone through. Just want to talk about my journey, probably give you some feedback, some insight. So, as Winkler mentioned, I've done a lot of different things, and I'm trying to figure out what to talk about or what to kind of focus on. And I realized that, a lot of seminars I've been to over the past years, most of the times it's just, some random person talk about something that, I don't really care about, or it doesn't impact me in any way. And I don't want to do that. Like, I don't want to be up here for an hour just telling you random stuff. I really want to make, go home with a message, or, learn something, or, I'm curious what you guys are into, what you want to know, and just how my journey can inform you in any way. So the more beneficial seminars I've been to is when I could just walk up to somebody and say, like real talk, how did you do this? Or what was your experience in this? What did you have to go through? And, that's kind of the tone I want to set for this, so just be as, informal and casual as possible. Like, if you have a question, please don't be shy. But I really just want to run through, my experience, and hopefully can provide some insight in any questions you have. So I can share my screen. Just load up my presentation, one sec. Just let me know if you can see it. Good. All right, awesome. So my title, good, okay. So my title is A Mighty Journey Through the STEM Field. Saw what I did there. My last name, I just kind of snuck it in there. But I really just want to talk to you about my journey, just kind of give you some insight, as I mentioned. So as I mentioned, we can go any direction you want. I've done a lot of different things, and I really want to hear from you and try to understand, like, what would you like to learn about or hear about? So to give you some structure, I kind of bucketed my journey into kind of three big buckets, or kind of three big pieces, how I see it. So I broke it down into kind of my research career, my thesis work, and different career opportunities I've had throughout my lifetime. So I've done a lot of different research in different parts of the world. I started in research maybe around in 2007 or so. I graduated I left San Diego, came to New York in 2006. Anybody here from San Diego? I know my dad is, but he doesn't count. So I'm just wondering if there's anybody else from here represent. But I went to New York, and I had no idea what I was going to do. I did the sciences in CXE, and I was always passionate about biology. I wanted to do something in the biology field, but I had no idea what that meant. And like most Jamaican parents, they'll tell you, you should go into nursing. Nurses make a lot of money. That's pretty much what they tell you to do. It's a good choice, good career choice, but like I just wasn't into that. Like I hit hospitals, this wasn't for me. So I wanted to kind of explore, what else I could do in biology, besides just being an instructor or something like that. So I really had no idea, and then I got into research, and I totally loved it. So what I have here, just kind of bullet points, just kind of highlight different things that I've gone through, and kind of trying to gauge, what people would be interested in, and kind of, give some feedback of what you want me to kind of focus on. I'm happy to run through everything, but if there's anything that stands out, just let me know. So, I started doing research in a stem cell lab in my undergrad, and the point of that lab was to try to use stem cells to repair damaged neurons in the retina, so try to restore vision from damaged photoreceptors. I did that for a few years, and that became the basis of my thesis work, looking into more a clinical approach. I also did some other side projects, some really cool things for a couple years, and published a couple papers. I looked at the effect of plant extracts on inhibiting growth of tumor cells. So we primarily looked at extracts from black cohosh, primarily actine. We looked at extracts from rosemary, from different extracts, compounds. And I know Jamaica has some extracts. I'm always curious, if you guys have some plants or something I could use to test this out. I know your grandma told you some bush from some tree that's going to cure this or cure that. So, always interesting to learn what that could look at. But I did that for a few years, and happy to walk through that research experiment. I also did some work abroad. For like 4 summers, I was traveling to Europe to do research. When I was in Austria, I was primarily doing stem cell research, but for a summer I did some bacteria work. So I worked in a bacteria lab looking at microbial volatile organic compounds that can act as agonists for pathogenic bacteria. So the basic concept is that I can take good bacteria, look at what they're releasing and how they can actually use this to fight or actually destroy pathogenic bacteria. So that was the thesis project I was working on there, and I have some cool data to show you there. Pretty exciting. But then, majority of my work was based on my thesis, and for my thesis work, I was trying to find early biomarkers for diabetic retinopathy. It's pretty much how you lose your vision from diabetes. So for a couple of years, you look at that picture, that's a picture of human eyeball. I used to cut up human eyeballs for a couple of years, and I also got patient urine samples from diabetic patients. So if you want to see some stuff about me cutting up eyeballs, I'm happy to show you that as well. But the basic concept for that is, one of the worst things in diabetic retinopathy is early detection. By the time people figure out they have diabetic retinopathy, it's too late and the disease has progressed too far. So one of the things we're trying to implement is trying to establish early biomarkers and seeing if we can use that from biological fluids, such as, finding proteins inside your urine. So what I did pretty much was take healthy patients, disease patients. I got urine samples from both, and I got eyeballs from healthy patients and patients who had diabetic retinopathy, and then looked at the expression profile in both healthy and disease populations to try to find any markers that could early signals for detecting the disease. So that's what my thesis work was based on, and I'm happy to walk through that. And then finally, the final bucket are my career opportunities. So, I've done a lot of different things throughout my lifetime. As I mentioned, I knew I was interested in biology, but, I didn't know what I wanted to do. I worked in the lab for, about 10 years or so, really loved it, but really wanted to explore other options, other career opportunities. And I would go to these seminars and talks and try to figure out like, what could I do just beyond going in academia or going in industry, and really kind of open my mind to the different opportunities that are out there, in either like government or scientific policy or scientific writing, the investment firms. So I had a few stops along the way. I worked as a consultant in life sciences for a couple months. I worked at an investment firm evaluating pharmaceutical companies for a couple months as well. I got offered a job as a medical communications expert, looking at the scientific writing. And for a couple of years, I was also a program coordinator for the Lewis-Stokes Alliance for Minority Participation, which is primarily focused on increasing the numbers for underrepresented minorities engaging in STEM fields, so science, technology, engineering, and math. Because you look at the numbers, it's a really, huge discrepancy in, minorities engaged in science and actually engaged in graduate degrees and actually having careers in these fields. So my role with that program was to try to, recruit students, recruit minority students, try to get them engaged in research and actually get them to go on and pursue graduate degrees, including master's and PhD. So, this is kind of a snapshot of everything I've gone through. And, if there's something you want me to focus on or something that's more interesting, please let me know. I want this to be as interactive and, low key as possible. But, these are kind of things I want to talk about today. But if there's any feedback or thoughts on it, just let me know. Or if not, I can kind of jump right into it.
Dr. Mikey.
Yeah, man.
Hello?
Yep, I can hear you.
Greetings, man. Glad to have you here.
Blessings.
Quick question. Can you talk a bit about your journey from, high school and then going to the research field? Like, what subjects did you do, and how did you transition to the university system abroad?
Yeah, definitely happy to talk about that. So when I left San Diego, I took chemistry, biophysics, geo, social studies, math, English A, and English B. I think those were my subjects. And, I left Jamaica in like 2006. And, I think my situation is a bit different. And I don't want to just kind of gloss over it. I was born in Florida, so it's a lot easier for me to just get up and go to the United States. And I understand that might not be the same situation for everybody here. So my experience might not be the same thing for everybody else. But just talking about my journey, I knew I could do that. So that was my plan. And, foolishly, I went there in, I think I left in like August, and I went to like a university and said, hey, I want to start school here in September. And they looked at me like I was stupid. I was like, what the hell are you talking about? No, that's not how this works. So I had like a year where I was just like, trying to get all my documents together, and I had to get like my CXC grades and stuff like that. I had to take like entrance exams. So, I left in August. And September, I think I got a job at McDonald's. I was working at McDonald's for like 6 years or something while I was in undergrad. But, I got to Lehman College, and I started, like I mentioned, I think my mom said, you should do nursing. Nursing makes a lot of money. And, I started looking into that, some form of biology and anatomy and physiology is interesting. But what happened to me is like, I wasn't exposed. I didn't know what a scientist was. I didn't know what research was. I mean, I've seen it on TV. You've seen scientists, you know what they do, but, an actual example of that, wasn't very prominent in my mind. So it was just random. I was taking a class with this guy who recently came from Harvard. He just started as a professor there, and he was looking for students to join his lab. And I was doing good in his class, and he asked me if I wanted to join his lab, but, I had no idea what that meant, or what, I thought he was just doing like some lab in class or something. But it's really cool because I got to see, how you go from scratch and actually build a lab and actually understand, what the lab is going to be doing, and just understand his research and background, and actually seeing an empty space transform into, getting a microscope, getting a cell culture room, actually getting cells in, learn how to pipette, learn how to culture cells, and just seeing that whole process and actually having an idea and executing on it and kind of developing specific aims and actually going through the experimental process of trying to answer those questions. So that was very engaging and interesting for me. That kind of hooked me from there. And then I just started doing different projects, from doing stem cell research to cancer research, like microbiology, and just trying to get my feet wet and just trying to see what else is out there and just different possibilities. And, I was fortunate because I was able to join a bunch of different programs. Somebody came up to me and said, hey, you're doing research, do you want to get paid to do research? And I was like, wait, what? Like, for real? So, I was like, yeah, sign me up. So there's a lot of programs that were out there that, especially from minority students, try to increase those numbers and get people involved. But, beyond that, what was beneficial for me was it was important to see people who look like me actually doing research and, people doing their PhD. I never thought at all like, I could do a PhD. Like, when I lived in Jamaica, if you asked me, if you told me I was going to do a PhD, I told you no, hell no, what are you talking about? I don't even know what that is. So, what happened with me is that, we were in these programs and we were able to see like, graduate students and people doing their PhD. And I realized like, they came from the same place I came from, we kind of had similar experiences. So it just allowed me to understand like, that's a possibility and it's not something out of reach. And like, you can actually do these things. You can do a PhD, you can actually do research and get funding and get grants and stuff like that. So it's something, very tangible and something more of a community and understanding like, there are people out there who are trying to help you and open doors for you. And that's why this kind of created this path for me where I started doing this kind of mentoring and support and trying to get students like me from minority groups to actually engage in research as well. But yeah, that's kind of the path. And I figured I want to do something clinical for my thesis work. So that's why I started looking at actual disease and start looking at something that can be translated to the clinic, something that could be actually applied in the clinic. And that's how the development of my thesis work actually came about. So, that's kind of a snapshot of kind of how I got into research and just, how I actually came to where I'm at today.
All right, cool. Thank you. Thank you so much for that.
Problem.
Seems almost serendipitous. I mean, what if you had not met that particular gentleman, would you be at McDonald's still,
Probably. They wanted to turn me into a manager too. I almost took the exam. I was like, nope, sorry, not doing it. But yeah, so, that's a good point. I think it's just, I was ignorant of these things. I didn't know that these things existed. And, that's why I think it's important for us to kind of, publicize these things and kind of, talk about these things so people are actually aware of all the opportunities. And there are people out there trying to help them and trying to pave a path for them. So, it's something passionate I really care about and, trying to make a difference about it. Cool. Any other questions or anything you want to hear about? Do you want to see eyeballs? Do you want to see cancer cells? What is it that you're interested in seeing? Or just want me to just go crazy?
Yes, why not? Let's go ahead with that. I mean, all right. That eyeball was kind of eerie, but yeah, let's.
All right. See, I'm just because of that, I'm going to jump to the eyeballs just because you said it. All right, so anyway, let me just run through my research real quick. I'm just gonna hit the highlights. So times it. All right, 20 minutes in. All right, cool. So as I mentioned, I did a lot of research in oncology, trying to evaluate how compounds from plants can actually inhibit the growth of cancer cells. So one compound we actually looked at is this from this plant called black cohosh. You may have seen this in like a drugstore or something. It's normally used for anti-inflammatory properties. I think it's also good for reducing symptoms of menopause in women. But one of the active components inside black cohosh is a compound called actine. And I worked with this lady who's actually evaluating these compounds, and I was able to kind of assess how they actually affected cancer cells and actually inhibited the growth of cancer cells. All right. So I wanted to understand the mechanisms of how this actually happened, and this is a very simple experiment called calcium imaging. So calcium signal is very important for pretty much all the mechanisms inside your body. If you look at a mechanism, calcium is going to be somewhere in there, using it as an extracellular messenger to communicate something in the metabolic pathway. All right. So what I want to understand is if I expose these cells to actine, what happens to the calcium signal inside the cells, right? So what you can simply do is you have a fluorescent dye that you stain these cells with, and then you can actually measure the level of calcium that's being excreted or released from these cells. All right. So this is before and after stimulation. So at this point right here, this is just measuring the calcium levels. All right. So what's happening is that this scale right here going from purple to red is from low to high. So as you go from this greenish to red, this is an increase in calcium happening. So at this point where I stimulate these cells with actine, you can see the increase in calcium being released inside these cells, right? So that's one part of the mechanism of how these cells actually are killing cancer cells. They actually release calcium and actually cause the mitochondria to release a protein called cytochrome C. And then this actually causes the cancer cells to actually die, all right? So I wanted to look at that and actually see how this is actually killing the cells from microscopic level. So I actually did this on HC29 colon cancer cells and these MCS7 breast cancer cells. And basically what I did this kind of titrated assay looking at different concentrations of the drug, as well as looking at it with combination of a compound that actually affects the cell growth pathway, just to see if there's any synergistic effort with that. So this is the control cell population right here. So this is just what the cells look like under normal condition. And if you expose them to 5 micrograms per ml and 30 micrograms per ml, you can see as you increase the concentration, these cells are going to start to die. And you can also see this inside the graph as well, looking at these cell proliferation. So as you increase the concentration of the cells, you see increased cell death as you go across the concentration gradient. All right, so this is something we're trying to evaluate. If we can use this and actually figure out what concentration optimally actually kills the optimal amount of cancer cells. And the cool thing about this is that these compounds don't affect normal cells, they specifically target cancer cells. And it's really cool because they release calcium to stimulate cytochrome C release, which kills the cancer cells, but then it also causes cell cycle arrest. So if you look at tumor cells, so cell growth goes to like a checkpoint where like cells are going to replicate, then stop, replicate, then stop. And there are checkpoint markers that allow them to go through that. But in cancer cells, these checkpoint markers are broken. So these cancer cells just replicate and keep replicating, and that's how they develop tumors, right? So the cool thing about this drug is that they actually cause the cell to go in a cell cycle arrest, so it stops the checkpoint and then actually causes cytotoxicity and kills those cells. So this is a really cool experiment that we did, and kind of this is what we kind of evaluated for a whole host of different compounds, just seeing how they target different types of cancer cells and us trying to understand the mechanism of how this actually happens. All right, so this is a project I did a couple of years ago in my undergrad, but thought it was relevant because, you know, I hear about all these amazing compounds that, you know, from this bush or from this leaf or from something. So, you know, excited to, you know, see if there's anyone who, you know, has any insight into anything they want to test out or think that they have something that, you know, I'm pretty sure, you know, your grandma told you something that can kill cancer, drink this tea or something. So this is just something I find interesting here. But let me stop there and just see if there's any questions around any of this.
You have a structure for the compound, the actins?
I do. Forget I'm talking to chemistry majors, so I didn't put it in the PowerPoint, but I do have the structure. I can actually pull it up afterwards and show you. I forgot I had to show all these structures. All right, but I can pull it up at the end of it just to show you if you're curious. But let me focus on something else, I guess more chemistry focused. So this is a project I did in Austria, all right? And this is a really cool fellowship I did. It's called the Marshall Plan Foundation. So basically, they fly you out to Austria for about 3 months. They pay for your flight, they pay for your boarding, they give you a stipend to survive. But for this project, I was trying to characterize microbial volatiles. So these are just compounds released from different microbial bacteria and fungi, and trying to see how well they can inhibit or stop the growth of pathogenic bacteria like E. coli or Staphylococcus aureus. So it's basically using good bacteria to try to inhibit or kill bad bacteria, understanding what particles are being secreted to actually cause this cell death, all right? So pretty straightforward. What I had to do was kind of evaluate a whole host of microbes, around 358 or so. And this is an example of what you want to see. So this is 2 different types of bacteria. You can see how the bacteria looks normally cultured right here. And when you put a antagonistic bacteria secreting some substances that's going to stop this bad bacteria from growing, you get what's known as a zone of inhibition. So basically what this means is that this trying to get my pen. It means that this bacteria is secreting some type of compound that is stopping this pathogenic bacteria from growing, and you get this zone of inhibition. All right, so what I want to do for this project is actually figure out which bacteria are actually antagonistic against pathogenic bacteria and figure out what molecules are actually secreting and how we can actually utilize that. All right, so I grew or cultured a whole host of different microbes, from bacteria strains, different fungi strains. You can see a couple of them here. And then I set this very simple experiment to understand how we can see what type of molecules are being secreted, all right? So it's a very simple experiment where you got 2 6-well dishes, And then you have pathogenic bacteria cultured on these textiles, right? So you put a drop of the pathogenic bacteria on these textiles, and then on the other 6-well plate, you'd have, this is the good bacteria, good fungi, right? And what you have are these holes or these pores inside this film that allows for the volatiles to be secreted from the good bacteria onto these textiles with the pathogenic bacteria, right? So what you then do is just kind of clamp this together. You put the good bacteria on top with the pores to allow for the volatiles to be secreted, and the bad bacteria with the textiles on the bottom, and leave this for about 48 hours, right? And then what you would do is then take those textiles and just kind of tap them on the agar and see which bacteria is actually inhibited in growing, all right? So an example of this is like, this is the control when there's no good bacteria in the environment, and then this one is exposed to an antagonistic or potential antagonistic bacteria, and you can see the reduction in the growth, right? And another example is looking at this. This is your control group. This is what the bacteria grows normally under the conditions, and this is when it's exposed to a potential antagonistic bacteria, all right? So after we screened all of these, we then kind of figured out which ones are actually strong antagonistic samples, so which one are actually inhibiting the growth of the bad bacteria, and then figured out what compounds are actually being secreted through mass spectroscopy, all right? So I'll stop right here so you can take a look at this. I'm a biologist, so this looks all like a lot of numbers to me, but I'm pretty sure you guys are all excited about this. I mean, I get the concept of what this is, but I'm pretty sure you have more insight into what these mean. But basically, we kind of evaluated, did some mass spec on all these potential antagonistic bacteria, and just tried to figure out what organic compounds are being secreted, and then try to categorize them. All right, so from the bacteria that we actually surveyed, we got around 16 compounds that we could identify, and then we classified them into these kind of larger buckets. So we found a couple antimicrobial compounds, some antioxidant compounds, and also some anti-cancer compounds, all right? So then from there, we can then kind of use these good bacteria and actually use this kind of mass produce these compounds that we can use in industrial manufacturing. So this is what the actual project was about, and this is what we kind of use this for. So you asked for structures and stuff, so let me just stop there and see if you have any questions about this one.
Okay, students, do you have any questions or any comments? Asking grad students initially, and if there are any other persons who have any questions or comments thereafter.
All right, no worries, no worries. I can keep it going. I just wanted to bring in some chemistry aspect of it. Just don't ask me to explain it.
In terms, I mean, one of the things that I was fascinated with a while ago is that you said you screened 358 microbes. Yeah. And this was over a summer. This was a short time period. How were you able to go through all of that? That was question one. And question two is, you speak about this Austrian Marshall Plan Foundation. How, I mean, I don't know, you're in the US and it's a different scenario from us here in Jamaica, but how did you get that fellowship award?
Got it. No, good question. So the cool thing about microbes is that they grow like incredibly fast, like ridiculous. Like you can culture bacteria overnight, and like you can screen like multiple amounts of bacteria overnight. So I think it took me a month to do like initial screening. So they gave me like a whole batch of fungi and bacteria that they were evaluating. But it's really cool because I used to work with cells and cell culture. You know, different cells can take like, you know, a week or so to actually have a full culture. But with bacteria, you can, you know, put some agar on it, drop it on a plate, and the next day you have like a full set of colonies. So it was really high throughput screening that you could do, which is really cool and really fascinating for me because, you know, I used to just work with cells. I never used to work with bacteria. So this was pretty exciting for me. So that's how I could actually get through it. So it took me like a month to just kind of identify the ones that I wanted to kind of focus on. And then the rest of the time actually doing the screenings and setting up the experiment. With the Marshall Plan Foundation, it was through the LSAMP program that they kind of brought it up to me. The year prior, I went to the Netherlands, and then so that group had like a bunch of different countries that they would explore through the City University of New York system. So it's not limited to United States, like there were people from all over the world in that program. I think the basis is just kind of identifying a lab that you want to work on and having a project. But I think they're open to, you know, different places across the globe. It's not just limited to the US, but it's a very cool program. I think when I did it, there are certain limitations that you just had to be there for 90+ days, and you had to stay at either the Graz University of Technology or the Austrian University. So the hardest part was just finding a lab to work in and having them sponsor you to actually be there for the summer. But yeah.
Okay, thanks. You can go on.
All right, awesome. So let me keep going. And I know people want to see eyeballs, so I'm just playing, but let me jump to it and just kind of get what I did for my thesis work. I did drop this in there just as like a humble brag. I'm just playing, but through this, I was able to do like a lot of cool stuff. You know, I went to Europe like 4 different times doing different things, conferences, scholarships, but you know, with that, it was really cool. You could travel across Europe for the summer. So this is just, you know, a couple pictures of me and all over. So I did research in most of these places, but it's very fun, very exciting. So, you know, I think it's definitely opportunities you should take advantage of. And, you know, happy to kind of talk through any opportunities, or if anybody's interested, just how, you know, how we could probably look into this. All right, cool. So a lot of my work, you know, I did around my thesis project. And as I mentioned, I used to work in a retinal lab trying to understand how to restore vision or different clinical diseases that affect vision. So for my thesis work, I focused on diabetic retinopathy, and more specifically, understanding the signals that are sent through these particles known as extracellular vesicles. All right, so I'll keep this very high level. So if there's any questions about it, just please let me know. So just a very simple explanation of vision and the retina. So what you see or what you interpret as vision is just simply light. So there's photons actually exciting cells inside of your retina. So these are your rods and cones. So these cells are actually going to take these photons and actually convert them into electrical signals. And then this is going to go to your brain. And this is what you perceive as vision. All right. So the cells that are responsible for this are known as photoreceptors. They're divided into rods and cones. So rods are more used for low light, and cones are more used for vision and acuity. So an example of that is like rods are responsible for showing like shape and structure. So if you look at a clock, your rods are going to allow you to see that it's a circle or whatever shape your clock is. And your cones are more for crispness and acuity. So your cones are actually going to allow you to see like the numbers on the clock, right? So this is how your vision actually works and how these cells actually play a role in that, all right? So when you actually lose your vision, what's happening is you're getting degeneration into these cells, which are your rods and cones. And in diabetic retinopathy, just the elevated levels of sugar inside your blood are actually going to affect the blood vessels inside your eye and lead to a whole cascade of problems, which I can run through right now. So the reason why I chose this is that diabetic retinopathy is kind of the fastest kind of growing neurodegenerative disease of the retina in the world, and also very prevalent in the United States. So I have this image here, and I had this image like 5 years ago, but I kept it because it's still very relevant. So this was a projection done earlier. This is the numbers for, these are numbers for diabetic retinopathy. In 2010, it was projected around 7.7 million. And by 2050, they projected it would be around 14.6 million, all right? And these are the actual numbers in actual 2020. So this was done in 2013, where it's already exceeded the actual amount. So this just shows you how rapidly it's growing. And then I looked at this yesterday, and the actual numbers for that retinopathy currently in 2020 were around 32 million. So this just shows you how fast this is growing, and this is the leading cause of blindness in the working population, right? So something very nasty. Diabetes affects every single organ inside your body, but the more kind of vulnerable ones include your kidneys and your eyes, just because of the dense capillary network inside these organs, right? So because of that, I wanted to focus on diabetic retinopathy and try to, do something to help in diagnosing this and use this for early detection. So that was what my project was based on. But just what happens in diabetic retinopathy, obviously this is a disease caused by diabetes, which causes elevated levels of sugar inside the blood. And what this does, it actually damages the integrity of your blood vessels, right? So there's a dense network of blood vessels inside your eye. If you look at your blood vessels, they kind of fit like Lego pieces, and you have like these protein structures that actually cause them to kind of bind together and kind of create this architecture and structure. So what's going to happen is the elevated levels of sugar is actually going to damage that integrity. And what's going to happen is that your blood vessels can actually start to collapse or break down or start to leak. And this is going to cause a whole host of problems inside your eye. All right. So a couple things that could happen is that your blood vessels can actually collapse just because the integrity and the architecture is damaged. And this causes aneurysms or swelling inside your eye. And they can also break and leak and cause hemorrhaging or leaking fluid inside your eye as well. All right. So this then causes a whole cascade of problems. And diabetic retinopathy kind of occurs in two different stages. There's the non-proliferative stage and then the proliferative stage. The difference is that this means that new blood vessels start to grow in the proliferative stage, right? So what happens is as your blood vessels get damaged and they start to damage the integrity of your blood vessels, your body tries to grow new blood vessels. The problem is that these are very brittle and weak, and then they kind of break down and form scar tissue. And as the scar tissue is formed, it actually pulls the retina from the back of your eye, and this is essentially how you lose your vision from diabetes, right? So something very catastrophic, very bad. But the problem is that there's no real form of early detection. The only time you know it is when you actually go to the doctor, when you start losing your vision. And if you're honest about it, like, when's the last time you got an eye exam? Like, no one goes to the eye doctor until something bad has happened, they start losing their vision. All right. So what we wanted to do is actually create a technique or a method that we could use for early detection, a very non-invasive way, just looking if you could use biological fluids such as your urine or your tears or your saliva to actually figure out what signals are being sent, and then possibly use this to try to identify markers we could use for early detection. All right. So the way I did this is that I use these particles known as extracellular vesicles. So as cells communicate, they're sending these tiny particles, and inside these tiny particles are different messages. There's different gene signals, protein signals, metabolite signals, and this is essentially how cells are going to communicate. So the hypothesis was, if in a disease state, there's going to be messages sent that are either going to increase the disease presentation. And what we could do is then kind of capture these messages being sent in a disease state and figure out what that message is, and then use that as a early detection for diabetic retinopathy. All right. So the whole premise of this is actually isolating these extracellular vesicles from diseased tissue and comparing them to healthy tissue and figuring out what the difference is between the two populations. All right. So how we actually did that and what the kind of larger question was is, do these extracellular vesicles play a role in diabetic retinopathy pathogenesis? All right. So there's a couple of experimental methods that we actually went through. So the first thing we had to do is isolate the retinal tissue. So I got eyeballs from cadavers. So I actually had an agreement with the New York Eye Bank, and it was very stressful because I would wait for, unfortunately, people to die, and if there were donors, then I could get their eyeballs. So this can happen at any time. I remember I was sleeping one night at 11 p.m. and someone called me yep, somebody just had a car accident. You need to come get their eyes. So I had to hop on the train, go all the way downtown to pick up some eyeballs. And it's really weird when you're on the train with a biohazard sign, a box, and people are just kind of looking at you wondering, what the hell does this guy have? So this is how we get eyeballs from cadavers. Also, people would ship eyeballs to me from different places across the states. But the first thing I need to do was get healthy patient eyeballs and diseased patient eyeballs, and then isolate the retinal tissue from that, right? So when you actually get the eyeball, what you need to do is kind of remove the fibrous layer, so the scleral layer, the white part of your eye, remove that fibrous tissue. And then essentially what you're doing is then kind of peeling the eye back into this kind of 4-leaf clover structure. And then essentially what I want to do is remove the neuronal layer and remove the layer that has the retinal tissue actually inside of it. So this is me peeling back the layer of the tissue and separating the retinal tissue from the fibrous layers inside of your eye, right? So once I actually had the neural layers, then I'd actually culture them for a couple days or so. So I'd actually take the tissue, put them into medium, culture them for about 5 days or so. And this would allow all the particles from the retinal tissue to be released in that media. And then from there, I would isolate that medium and then go through ultracentrifugation. And this just spins it super fast. And this would actually allow me to isolate the particles that are actually inside that media, right? And then from there, what I did was actually break up those particles and just try to figure out what the message is inside of those particles, right? So I did a couple of experiments. Nanosite is a technique used in light to look at the actual particle concentration. I actually did some microscopy on the particles. We did some next-gen sequencing, looking at the genetic profile of the particles, and looking at mass spec to actually look at the protein actual concentration and protein distribution inside the particles as well. Hoping that we could then compare the healthy tissue and the diseased tissue and see what the difference is between those profiles, and hopefully find some markers that were only specifically in the disease profile. And then we could use that as a marker for early detection. All right. And then from there, we did some bioinformatic analysis, understanding what these genes and what these proteins were actually responsible for. All right. So just the kind of quick summary, what we actually identified. So these were all the upregulated proteins inside the diabetic samples. So the hope is that, if you look at the tissue is going to release in these EVs or extracellular particles. And what we're hoping for is that these particles are going to contain some signal that can be used as a biomarker to detect that this person is developing diabetic retinopathy, all right? So what we did was look at the upregulated proteins that are released from the tissue and that are also present inside the retinal EVs. And we found 3 unique proteins, or 3 unique actual components. And the 3 of them were an XNA1, ceruloplasmin, and LDLRP1. And just looking at the functions of what these actually are responsible for, this is responsible for neuroprotection. So upregulated, this actually helps to inhibit inflammation. So this makes sense in a diabetic retinopathy patient. There's high amounts of inflammation, so this protein would be upregulated inside this. There's also ceruloplasmin. This is also responsible for elevated levels inside someone who has diabetes, observed in both type 1 and type 2 diabetes. So this is your diabetic environment representation. And then we have this LRP1, which is responsible for angiogenesis. So this is the new blood vessel growth that happens in proliferative diabetic retinopathy. So these proteins that were found were all kind of responsible or signifying to this disease presentation, this representation of diabetic retinopathy. So this is something that we found that we thought was very interesting and very relevant to us actually finding markers that we could then isolate from the media and use that as actual markers to detect if someone is developing diabetic retinopathy. All right, and just kind of the punchline of something else that we did. So, we were trying to do this in a non-invasive manner. So the hope was that, you could go to the doctor, you could pee in a cup, and I could then take the microvesicles or extracellular vesicles from your urine, look at these signals and tell you if you actually have one of these markers and kind of give you a flag to say, hey, because of these markers we found in these concentrations, you're at risk for developing this disease or this profile, all right? So what we did is that we looked at the vesicles released from the tissue and also the vesicles released from patient urine. So someone who has diabetes, someone does have diabetic retinopathy, what signals are being sent from their retinal tissue and what signals are actually found inside the urine? And can we isolate their urine and actually find the same signals and actually predict if they actually do have diabetic retinopathy based on those signals, all right? So these were all the upregulated shared proteins that we found in diabetic patients from their urine and their retinal EVs. And we found 43 proteins that we then used to kind of characterize this as like a profile to detect the patients. So if you have these levels of proteins inside your urine, this could signify that you are progressively developing diabetic retinopathy. And these are all elevated when compared to healthy patients. So this profile of proteins that we found was kind of the markers we used to kind of tell someone, hey, if you go to the doctor, you pee in a cup, you have this expression of these proteins, this could mean that you have diabetic retinopathy, but this is just kind of something you would then use to kind of trigger them to do deeper tests, to actually do some imaging and do some fundus microscopy inside their eye to figure out if they actually do have diabetic retinopathy or not. All right, so that's pretty much what I did for my thesis in a nutshell, but pretty much, worked with eyeballs and urine for a couple of years. But I'll stop there and just see if anyone has any questions around what I just went through.
Any questions, guys? Any questions?
You guys are quiet. You're just nervous? What is it? I don't know. All right, so let me run through it. So that was my thesis work. Oh, you have a question? Please go ahead.
Yes, Dr. Mighty. All right. For someone who want to look at any program, any issues, do you have a structured way of taking a problem from just as a problem and move it through all the research stages to come to what do you do, what do you look for, what do you test for, and to an end result that gives you something like this?
Yeah, so currently I work at Regeneron Pharmaceuticals, and we have really a drug discovery pipeline, right? So basically there are different departments that deal with oncology, deal with muscle metabolism, neuroscience, ophthalmology, and they're all dedicated looking at different disease profiles. So specifically I work at the Genetic Center, and we're looking at how genes actually play a role in disease presentation, right? So basically you can go sequence your genome, and you can look for mutations inside your genes to say, okay, if I have this mutation, that means that I'm at risk for developing this disease or not, right? So there's a whole pipeline of different experiments that you can do. So, let me speak from my perspective at the RGC. So let's say, we sequence 100,000 people, 100,000 healthy people and like 10,000 people who have Alzheimer's, right? What you're gonna find are signals that are associated or mutations inside your genes that are gonna signify inside the disease population that these mutations are linked to Alzheimer's. What you can then do is then look at that gene and look at how this is actually expressed inside different patients, right? So you can take their brain tissue, you can look at like protein presentation, and then you can also look at different mouse models. So ideally what you can do is you figure out if this gene is mutated, all right, and you understand that this gene is gonna then create this clinical phenotype. Like let's say you have a mutation in the gene, it's gonna lead to like increased cholesterol levels or increased intraocular pressure inside your eye. What you can do looking at a mouse model is actually knock out the gene. So you either knock it out or knock it in. So you can either insert the gene or knock out the gene and see what happens in the mice. So we've done this with things like obesity. So an example is there's a gene called GPR75, and this is linked to regulation of obesity. So what happens is if you have a mutation in this gene, you're more protected against developing obesity. And how they did this is that they had two population of rats based pretty much, and they put them on like a high fat diet. And the mice that had the mutation, they were fed the same amount. They developed less weight than the mice that didn't have the mutation. So this is how you kind of take, you look at a gene, you look at a disease, and you look at all these different characteristics. You look at the genetics, you look at the biology, you look at the mechanism, and then from there you can understand the pathway, and then that's how you can actually develop therapeutics or drugs to actually solve these issues and actually create drugs to help patients. So they're distinct pipelines set up to address like a multiple host of different diseases and different conditions. It's just to kind of do this data mining and understanding what the mechanism is and just how the disease actually presents itself. Yeah.
Okay, so question. So any person can go and get this profiling work done, and what would be the cost if one wanted to see what diseases they're prone to? And is that what is done with babies in utero?
Yeah. So you can actually, if you've heard of like the BRCA1, BRCA2 mutation for breast cancer, so there's a whole shift happening right now where people are taking clinical genetics more serious. So there's this thing about early detection where if I can sequence your gene and figure out mutations that you have and figure out if you're at risk for actually developing certain diseases based on the mutations inside your system, this is what's happening now in different health systems where you can actually go and get a genetic panel. And what they have is a list of genes or a list of mutations that are linked to like different genetic conditions or different diseases. So they have like a list of like, I think it's like 110 genes now that they know are directly linked to the development of different diseases. you can go and get this panel done, and then they can tell you if you actually have one of these mutations, and then start putting you on a regimen to start doing testing earlier. So like, if you have a mutation that might, lead to increased risk of prostate cancer, you might get your prostate checked earlier than 50 or so on and so on. So these are all different clinical representations that are used to actually engage patients earlier and put them on different treatments or try to identify the problem before it even starts. So you can actually go now and get a genetic panel done and actually figure out if you have some of these mutations. So if you look at like 23andMe and Ancestry and stuff like that, where like, they tell you your genetic makeup, and there's like a health component where they can tell you, you have, they can't directly tell you that, you have this mutation, so you have this disease. Just because of liability and stuff, they can get sued for that. So what they do tell you is like, you fall in like this percentage or category where you have an elevated risk of developing this or that. So you need to actually go to like a hospital system or health system who can actually do clinical sequencing and validate this. And then you can actually have genetic counselors who walk you through your genome and say, we found this mutation. That means that you might have this disease or a greater risk of developing this disease and so on. So the cool thing about it is that sequencing technology has advanced so much now. Like you can sequence someone's genome or sequence all their exons, their protein coding regions for like $12 now or something like that. So like $12, something really cheap. So because the technology has advanced so rapidly and it's so cheap, more hospital systems are implementing this inside their regular regimens and actually using genetic sequencing to actually advise patients on different avenues that they could take based on their health profile.
Okay. I had no idea it would have been so expensive, relatively inexpensive to do something like that. Wow.
Yeah, if you asked 5 years ago, it was probably a lot more expensive, and it wasn't, as known, or people, if you like, people know about like BRCA1, BRCA2 mutation for breast cancer a long time ago, but before you could actually get that done, you'd have to like show proof of like, your mom or some relative having it, and that's would be the validation for you getting the test. But now it's so cheap that you can go to the doctor and be like, I want to get genetic sequencing and to understand what mutations I have in my body, and then what disease representation that could happen in the future. So yeah. That's what we're doing at the RTC.
Jason, I do have a question regarding what you just mentioned. I believe you're in New York State?
Yep.
Okay. How does the state laws keep up with the results of some of those tests? Because I could certainly imagine that some people would not want to do that for the fear of discrimination if some of those results can negatively affect their longevity, let's say in the workplace at a particular company. Is New York State protecting some of those results so that employers can't use that in an adverse way?
Yeah, definitely. There's like really stringent rules in New York State, especially around genetic sequencing. Like to actually, you have to be actually CLIA validated lab or genetic sequencing lab to actually able to do this. Like not every lab can just go and do genetic sequencing. And for pharma companies and like universities, they can only do research level sequencing. They can't do the clinical grade, so they can't actually validate this. But to your point, this is all anonymous. So like if we sequence samples from 100,000 patients, we have no idea who they are. What it is just like a random number from the hospital system that is giving us their sequences and then give us their actual phenotype or electronic health records. But we have no idea of actually figuring out who these patients are. And this is all done through consent. So you can actually consent to have your genetic data be used for research, and they kind of walk you through everything that your data can be used for, how you want to use it, what you want to include it in. So you have the power as a patient to actually include your data. But there's no way of people who are actually interacting with your data. Like if you do like a 23andMe experiment now, like there's no way that we can figure out like it was this person, that person actually did this. So it's totally anonymous. There's a lot of HIPAA regulations and privacy and legal regulations around this. So there's no way that this can be used against you. What happens is that you have a relationship with the hospital system, and then you can go in and meet with your doctors and say, okay, I want my profile to be examined. And then you have genetic counselors who walk you through what this looks like. And then you can then make informed decisions about what your actual disease profile looks like. So it's a very interrelationship, but this is all very regulated, and it's like nearly impossible to actually do this in New York State. Most companies go like outside of New York to actually do it because the laws are actually a lot less stringent, but there are very tight regulations around this that don't allow you to just do genetic sequencing in any way.
Thank you.
Okay. Awesome.
Right.
Well, we are coming down to the hour.
All right,
So,
I got 2 minutes. Last thing I'll
yeah.
show you what the RGC actually does. So we actually collaborate with institutions all over the world, So we have collaborations with over 30 countries in the world, and today we've sequenced about 1.6 million patients. The whole point of that is that we engage with hospital systems, different universities, and then we get DNA samples from all these patients. We sequence them as long as getting their electronic health record data. And then basically what we do is that we match genetic data with the phenotype or clinical data, and basically look for mutations that could advise you about different disease profiles. So for example, if you have a mutation in this disease and it shows that it leads to like increased cholesterol levels, we can then find out what gene that is and what the actual mechanism behind that is, and then use that to develop drugs to target that specific pathway or specific genes. So the RGC is looking to sequence populations all over the world, different ancestral distributions. As you can imagine, most of the samples we have now are European populations, just because these are samples that have been sequenced from European countries or European ancestries. It's just, it is what it is. These things don't really translate to minority populations easily, or just not aware. So what we're trying to do is get more samples from more diverse samples, because if you run a clinical trial and only white people are in the clinical trial, how the drug is actually going to impact people from African ancestry versus Asian ancestry versus European ancestry is going to be different. So being able to understand the genetic diversity of all these different populations is what's actually allowing us to develop unique drugs or specific drugs to target different ancestral populations and different disease states. So this is just a nutshell of what the RGC actually does. But yeah.
That is quite interesting. Okay, awesome, very much. Are there any final questions that anyone wants to ask Dr. Mighty? Any final questions? Dr. Minor?
Yes.
Hey, Dr. Mighty.
Oh, okay.
Thanks for your presentation. I noticed you said that most of the sequences you have done are of people of European extraction. So what's the plan to try and get samples from other races?
Yeah, no, that's a great question. So what the RGC is doing is we have collaborations. We're engaged with different biobanks or collaborations from all over the world. So right now we're talking with collaborators in parts of Africa, in Puerto Rico, and different parts of the Caribbean, people from all over the world, South Asian population, East Asian population, because sequencing is normally done with European populations, people from like the UK or Finland or these countries, and the bank samples or the data out there is strictly for European populations. So we understand there's a big discrepancy in that, and there's different minority profiles that can advise us on how to develop drugs. So we are actively engaging with partners all over the world to try to bring in samples, bring in hospital systems all over the world to increase the diversity of populations that are in our portfolio.
I see your potential cohorts. I see none for the Caribbean region, but I do see you in South America. None in Central America either.
So we are engaging with a Puerto Rico university right now, and there's a couple in South America. Happy to have a conversation about Jamaica if people want to go down that road. But we're always looking for new collaborators and new samples to actually work with. And this is a partnership, so we'll actually do the analysis with you. We'll go through the samples, show you how to do the analysis, and actually allow you to interpret the data, understand, what mechanisms and what pathways are being affected. So this is the whole goal of what the RTC is trying to do.
Okay, that's interesting. I'm sure our medical faculty and biotechnology center would have appreciated being a part of this talk.
I'm here to stay, no worries. I'm always open to talking.
Okay, no problem. Thank you so much. I do have a final question. So you spoke about the samples, the eye samples that you get from persons. What percentage of persons actually donate their eyes or donate body parts to science in the US? What percentage?
Yeah, I looked at numbers previously. I'm embarrassed to say this because I used to get samples from organ donors, and I'm not an organ donor. I think last time I renewed my license, they asked me, and I realized I wasn't, and I felt like a hypocrite because here I was doing all my research from people who are actually organ donors, and I'm just not signed up. So I looked at it, I think the numbers were somewhere around like 30% of people signed up for it, but for me, it was very specific where I had to get people who were either healthy, meaning I didn't have any adverse conditions, or people who had diabetes. So I was limited to a very kind of narrow inclusion criteria. But I think when I looked at it, or when I checked this out, around 30% of people in the United States are actually signed up as organ donors. That number might have changed in the past few years, but when I was doing it, that was the percentage that I was looking at.
30, as in 30%?
Yeah.
Wow, that's pretty high. That's pretty high.
Yeah. They like put it on a box you can check when you renew your license, at least in New York. So it's like an easy box to check. That's it.
Okay. I don't think we have any such box here in Jamaica for sure. Okay. All right. Well, this has been.
Just commenting on that last point, we do not have any such box. I know that organ donation is increasing in Jamaica, and we do have some individuals who have indicated in their wills, since we don't have any such easy way of donating their bodies to science. So perhaps that's something that the country as a whole need to look into and push.
Fair point.
Okay. Matter to ponder. As chemists, scientists, what say we? Okay, no problem. All right, well, I think that
Twinklet, just on that note of donating, I think you can actually get a form from the anatomy lab. If you want to donate your body, at least to your lab, for, I don't know, research or for the undergrads or whatever, there's a form that you can collect and sign up, because they would need the body very soon after you die. And there's a form, I don't know if it changed, but some time ago, I have a form from somebody who had given me probably about 10 years ago that they were interested in donating their body, and they had to sign up something, got it from the lab and thing, and gave me to keep. So my responsibility was, as soon as the person die, and the police, if it's a case of police or a hospital, I present it, and they will take the body and do what they need to do. I don't know if it's, well, I need to find out if I still have that responsibility. But there is some way that you can donate your body, but I don't know about, and I guess the same thing probably with parts, but I know you can donate your body to at least the anatomy laboratory.
Okay, wonderful. And as I said, matter for us all to ponder upon. All right. Well, that just leaves me to thank you so much, Dr. Mighty. You have spoken about a whole diverse array of aspects of your work and aspects of your life. And it really has been quite inspiring for us all. And, the concept of going to different places and having new experiences is absolutely critical, because there's so much that you learn when you're in different spaces, and you learn different cultures and all of these other things, and the nature of the networks that you can expand with in that context is really very key and important. So thank you so very much, Dr. Mighty, for joining with us this afternoon by now, and for all of those who are online. I see Mrs. Lindo. Hello, Mrs. Lindo. Good to see you online. I haven't seen you in a while, but awesome. So thank you so much, Dr. Mighty, and the rest of you.
Thank you for having me. Really appreciate it.
Awesome.
Right, take care.
So,